Hello everyone, welcome to the latest Novogene webinar, introducing Novogene Europe's clinical sequencing services. This is the first of our clinical webinars and follows on from our UK Sequencing Centre receiving its ISO 17025 last month. Before I hand you over to your host, I'd like to thank you all for joining us today for what promises to be a very interesting webinar. A bit of housekeeping before we start. So the webinar will run for around an hour with approximately 15 minutes for question and answers at the end. Please submit your questions via the Q&A button at the bottom of your screen. You can now upvote a question by clicking the thumbs up symbol next to it. We will try and prioritize the most popular questions. However, if we don't have time to answer your question, we will get back to you by email after the webinar. Talking of which, we will share the slide deck and a recording of the webinar via email. You can expect this on Friday. So without further ado, I would like to introduce you to your host for the webinar today, Marco Dovis. Marco, if you could pop up, over to you.
Hello, John. Thank you very much. I guess you can see my screen and my video.
All good.
Great, thank you. Thank you very much for arranging this new webinar. It's such a pleasure and an honor for me to introduce to Novogene Europe's clinical sequencing services. I'm very glad to be here with you. Thanks to all the participants to join us. But I'm not alone today. The speaker will be Dr. Aina P, and she will be later on with us. I will introduce her later. But now I would like to start briefly with a summary of Novogene for the people who still don't know the big community we have in the whole world. Novogene is a young company. We have just had our 10th anniversary last month, but we already offer full service next generation sequencing. As you know, almost every day there's coming new applications for sequencing, and Novogene is involved in all of these applications, from research projects to biopharma, and from genetic health to complex disease, oncology, or even infectious disease, such a hot topic today that we have with the pandemic situation. Fortunately, our European lab has been able to run all the projects during these difficult times without any problems. Here you can see a picture of our Cambridge Sequencing Center in the Bioinnovation Center in Cambridge. It's a brand new lab running several Nova 6000 and also having a supercomputing cluster on site that makes possible to do data analysis very fast and very extremely fast. Yeah. But our Cambridge lab is not the only one we have. We have 5 labs around the globe, and starting from US, we have in UC Davis, a lab for American clients, and also in Asia, we have 3 big labs. Singapore lab has recently accomplished the 10,000 human whole genome sequencing project, which we are very proud of. And in China, we have 2 big labs next to our headquarters. These 2 labs has amazing capacities thanks to our new intelligent delivery platform called Falcon. You can see this robotic arm doing the whole process without any intervention of humans. And you can check more details in our website if you want, or in our social media. In any of our labs, we offered a wide range solution, genomic solutions, such as a brief summary we can see here. Starting from transcriptome services, we can check messenger RNA or even non-coding RNAs, including long non-coding, microRNAs, or circular RNAs. Apart from that, we can even go deeper into single-cell RNA-seq profiling or isoform resolution for these studies. As well, animal, plants, and microbial genomes have no secrets for us. We can resequence the species that already have the reference genome, or even create de novo reference genome for you, if needed. As well, for microbial genome projects, we can check taxonomical diversity through amplicon sequencing, or even go further for functional metagenomic analysis through shotgun metagenomic sequencing. Last but not least, we can also discover information about your epigenome through whole genome bisulfite sequencing service. And we can check as well DNA or RNA with protein interactions through ChIP-seq and RIP-seq services. But today, we are going to focus on human genome, which is most relevant for clinical services. We are going to discuss all details for whole genome sequencing projects, whole exome sequencing projects, and some targeted capture panels. Collaborating with Novogene has a wide range of advantages, but here today, I would like to highlight two of them. These are quality and turnaround time. At Novogene, we understand how important is diagnosis and how important is to deliver high quality and the fastest turnaround time possible. That's why we've made all our efforts to obtain recently ISO 17025 in our Cambridge lab. Thanks to our high capacities and our incredible high standards for quality for all our services, we have been able to have more than 580 research papers in the top scientific journals. Moreover, we have a worldwide global partnership with more than 4,000 institutes, including companies, private companies, hospitals, and research organizations and universities. If you want to become part of our large community, you can find local representatives just next to you. We will be glad, sales team will be very glad to help you and assist you in the whole process, from project funding application to completely analyzed data delivery. We aim to have to offer you an outstanding customer experience, even in your local language. As I said, we are just next to you. So please don't hesitate to contact any of our sales representatives or get in touch through our social media. We have LinkedIn accounts and Twitter accounts. You can also drop us an email at europe@novogene.com directly. We will be very happy to talk about your project. So thank you very much for your attention. And now I would like to introduce Dr. Aina P. with you. Dr. Aina P. has been doing her PhD in the genetics department in the University of Barcelona in Catalonia in 2018. After that, she joined us in Novogene Europe and as an operations manager. She's been quickly promoted to supervisor and recently changed to technical support supervisor position in the clinical department. It's such a pleasure for me to have her here today with us. And now I will let her tell you all the details about our clinical services. Thank you very much for your attention. So Aina, please, you can proceed when you want.
Thank you, Marco, for this nice introduction. Going to share my screen now. Okay. Good morning, everybody. So as I was saying, thank you, Marco, for this nice introduction, and welcome to this introduction of Novogene Europe Clinical Services. I am Aina, Technical Support Supervisor for Clinical Services of Novogene, and I am going to give you an overview of our services. So the structure of the webinar will be divided in 3 different sections. So first, an introduction that will be followed by the clinical products and requirements. And I will finish by giving you the information of our clinical pipeline flow. So along these 10 years that our company existed and has been a leader in the next generation sequencing field, Novogene has earned its reputation being a certified and trusted partner. With this, we have achieved being an Illumina certified service provider and Illumina Genome Network partner. In addition, we are very proud to be the only precision medicine center of excellence in Asia, certified by Thermo Fisher Scientific. On top of this, we have accredited laboratories all around the globe, with CLIA accredited laboratory, standing for Clinical Laboratory Improvement Amendments, a CAP lab accredited by the College of American Pathologists, and also the recently accredited ISO 17025 laboratory. Therefore, as you can see, we can process your samples with the high standards of clinical grade sequencing all around the world with our laboratories. So as you all know, related to next generation sequencing, this field and its applications have been rapidly growing, and currently, next generation sequencing is also widely used and accepted for both hereditary diseases and oncology diagnostics. So our clinical services aim to support you and to address the numerous applications that clinical next generation sequencing can give answers to, such as cancer diagnostics or monitoring, which will allow to predict the aggressiveness or response to treatment and correlate to drugs that would work best for each patient. Also, it has its importance into pharmacogenomics to assess the drugs and doses for the patients. As well, it's important to for doing the gene expression profiling, and as well, of course, for assessing like different genetic diseases and tests such as prenatal newborn screening, or as well could be for carrier testing. And finally, also it can be used for other suggestions such as lifestyle or medication guidance. So we do different like NGS services, always providing a high quality data to allow our customers to provide an accurate diagnostics for these purposes in their preclinical or clinical research. So now moving to the next part of my talk with the clinical products. So here you have in the list of our most demanded services that are included, that are whole genome sequencing, whole exome sequencing, our clinical panels, which today I will focus into the oncology panels that we have. Then we have the RNA sequencing solutions and our bioinformatics solutions with our Novo Neoantigen 2.0. So beginning with whole genome sequencing provides a comprehensive collection of individual genetic variation interrogating the entire genome. And with our service, we offer improved coverage and sensitivity for the detection of structural and complex variants. Also, whole genome sequencing, of course, enables the identification of non-coding variants, such as pathogenic variations that disrupt the regulatory regions, also non-coding RNAs and mRNA splicing. And it also allows to do the reanalysis across multiple variant types to increase the diagnostic efficacy. Regarding the whole exome sequencing, this one, it targets the protein coding regions, and it focuses in a much reduced area of the genome with in between 1% or 2% of the region of the genome. However, 85% of the disease-related mutations are known to be found in this exome region. For this reason, compared to the whole genome sequencing and for the same amount of data, whole exome sequencing has the advantage of a greater sequencing depth and therefore delivers more effective data. And for the case of patients that would have genetic disorders, clinical exome sequencing plays an important role in quickly identifying these potential variants. So here now I will proceed with giving you an overview of our whole genome sequencing service. So we are proud to offer flexible data output and coverage. By this, what I mean is that I want to highlight that we offer the typical 30x or 50x coverage, but also we can sequence much lower, offering you low pass with lower coverages, which is an expensive solution for high throughput strategy for detecting genome-wide genetic variation. And this strategy normally can also be complemented with imputation or whole exome sequencing to have a deeper picture of the variants. Also, we are proud to have a very fast turnaround time of just 15 days from sample arrival into our facilities until we release the raw data to you. Also, we offer customized deliverables for our customers that include BAM files that are related to the mapping. So BAM coming from a binary alignment map, or also we can offer variant calling files or annotation. As well, we offer a safe and personalized data transfer, giving you different options such as hard disk, Amazon Cloud Link, or as well, we could transfer directly to your server. Finally, I want to highlight the high quality data we can achieve. So here you have an example of our performance. So for a 90 gigabase service, we obtain over 90 gigabases in general, and also mean that achieved of around 30x. For the base pairs covered at least 10x, it's over 98.5%. And this quality parameter, the Q30, is obtained over 85%. Moving to the whole exome sequencing, for this one, we also offer a flexibility and coverage for your data, and a very fast turnaround time of 15 calendar days from sample arrival until we release the raw data to you. For whole exome sequence, also we can offer multiple target enrichments. So we offer different options such as Agilent or IDT, and we can provide customized deliverables again to you as you require. Also, we offer this safe and personalized data transfer to you. And so if you give us your details and your preference, we will do as you require. Finally, in terms of quality, so our validated whole exome sequencing service is a very powerful solution and can achieve the high standards of clinical grade services. And so here you can see the summary with an example of a 12 gigabase sequencing with the Agilent capture strategy. So here you can see that the validated mean depth sequencing achieves over 100x with over 12 gigabases as average raw data. And we can cover for more than 20x as base pair covered, over 93%. And in terms of the repeatability, sensitivity, and specificity for SMBs and indels, we can achieve over 95, 92, and 99% respectively. So having a very great performance for our service. Related to this, to be able to perform this whole genome sequencing and whole exome sequencing for you, here in this table, you have the requirements of the samples we need you to send to us. So for both services here, you have in these columns the requirements in terms of sample amount, volume, and concentration, the description of the quality preferred for us to receive your samples, and also our recommendations in terms of sample collection and transportation. In addition to genomic DNA and FFPE extracted genomic DNA, we can also accept tissue samples for us to do the extraction. And we have listed here the most common ones, such as whole blood and saliva, because these are the ones that are mostly used for collecting samples from a proben in a non-invasive way or in a minimum invasive way. So however, if the origin of your samples is from another type of tissue, or you want to, for example, use ctDNA for whole exome sequencing, you can always contact your sales representative or our clinical service email, and we'll provide you with all the details. Also, here in this table, we have listed the characteristics of the samples that we would require you to send. But if you cannot match our stated requirements, please also contact us, because we can assess whether we could still process your samples or give you an alternative so that we are still able to do and carry on your project. So moving to the next service, which are the clinical panels. These ones give you a very high coverage in clinical relevant genes. And so because it focuses in a much reduced area, also it can be very useful for both preclinical and clinical research. And Novogene has different validated panels. And today, as I mentioned before, I will introduce to our oncology panels in more detail later on. Regarding the turnaround time, it's again like 15 days only from the DNA sample receival until we release your raw data. Also, we can offer you customized deliverables that might include BAM files, DCF, annotation, and also tumor mutational burden or microsatellite instability. Also, we commit to safe and personalized data transfer, either by AWS, hard disk, or server transfer. and we provide very high quality data. So related to this, here you have listed our main oncology panels, NovoFocus CR, which is a cancer risk panel that focuses in 106 genes. So this panel is especially important for hereditary cancers and has many applications and use. So it is especially related to some hereditary tumors, as I said, like breast, ovarian, colorectal, uterine, among others. And it allows very much to assess the cancer risk and also the affectation and the origin of the mutations of one cancer that might have hereditary origin. Next, moving to our NovoPM 2.0, which is a solid panel with a tumor panel with 484 genes included. So for this other panel, we can detect several types of variants, specifically SMBs, indels, CMV, and structural variants. And we are very proud to announce that these are very important for clinical alterations. So as you can see here on this left hand side, we can see that NovoPM 2.0 not only comprehends the full number of genes that is included in Foundation 1 companion diagnostics, which is a very well known, recognized, FDA approved diagnostic test, but also many others that are also very clinically relevant for diagnostics. Also, I want to mention that the genes included in our panel are relevant for diagnostic or treatment of various solid tumors according to the National Comprehensive Cancer Network guidelines and the medical literature. Finally, I want to stress that these panels are done in our CAP accredited laboratory and are analyzed by our bioinformatics engineers in-house. So as I mentioned, we can provide you with bioinformatics analysis for this NovoPM 2.0. And here, let me introduce to you to the flow of our bioinformatics analysis. So once we obtain the raw data for your project after sequencing, and we perform the data QC to remove any low quality reads, we are able to get your clean data, and then we will be mapping against the reference genome. From this point, the analysis for detection of germline mutations can be performed for SMBs and indels. And also, if you send pair samples, we can detect also the somatic variants, somatic mutations for all types of variants, such as like SMBs, indels, CMVs, and structural variants. Also, we can perform the microsatellite instability detection. And in addition, the tumor mutational burden can be assessed from the data from somatic in SMBs and indels. So the outcome for this variant analysis is for great utility for different applications, as you can see listed here. So for the variants detected, we can be able to do target therapy based, and also do some recommendations in terms of immunotherapies based on the TMB, BTMB, or SMI scores. Also, it is very important for hereditary cancer risk assessment, because it can give you a more comprehensive tumor profiling for cancers of known origin and assess the efficacy of the chemotherapy. So related of this chemotherapy, moving to the next slide, as I was mentioning, to illustrate you better on how mutational detections on our panel are relevant biomarkers, you can see here different types of tumors and how the variants detected with our panels can be then used as biomarkers to use FDA-approved therapies to treat the H patient. So for example, you can see here for non-small cell lung cancer that having some ROS1 fusions could be treated with crizotinib, or in the case of ovarian cancer, detecting BRCA1 or BRCA2 alterations can be linked with the olaparib or rucaparib treatment. So to show you again, like the high performance of our Novo PM2 panel, I summarized the metrics here using the alterations detected with reference standards and also the ones detected with clinical samples. So with the reference standards, we obtained an accuracy of 99.5% and 93.7% for SNVs and indels respectively, and with a minimum allele frequency of 1%, and for samples with an average depth of 1,400x. Also, the detection rate of CNVs for HER2 is 100% when the tumor cell content is 20% or above, which is a very good detection rate, of course. And ROS1 and ALK fusions, our rate is detected in 100% as well, and highly reproducible when the tumor cell content is equal or above 20%. In terms for the clinical samples, we achieved really impressive results as well with 41 FFP samples, using them with samples with no prior mutations, with prior hotspots for indels or prior hotspots in fusions. And the results provided obtained an average sequencing depth with 1,400x, and were 100% concordant with the NMPA approved companion diagnostic test, and both for SNVs and fusion detection. So after having shown you the great performance and the metrics of our Novo PM2 panel, I will share with you now the sample requirements that are listed here in this table. So we accept different types of samples, including genomic DNA, FFP extracted DNA, or CTCF DNA. And as a reference for control, we usually use a genomic DNA derived from blood cells. So aside from these type of samples, you can also see the requirements in terms of total amount, concentration, and volume for each sample type, and also a qualitative description, which is basically we try to provide the samples without degradation or minimum degradation. And one of the most important things is, for instance, for CTCF DNA, that it doesn't contain any genomic pollution. Also in the table, you have our recommendations in terms of sample collection and transportation. And finally, after having mentioned that, in the table on the bottom, you have our recommended sequencing volume that we normally suggest. So 12 gigabases is suggested for the genomic DNA coming from tumors to achieve an effective coverage that in between 1,200 and to 1,400x. For CTCF DNA, we suggest at least 16 gigabases that would provide to you 1,400 to 1,600x. And for control DNA with just 1 gigabase, we are able to obtain 100x and use this data as a reference to compare your samples and be able to provide you with like somatic mutation calling as well. So moving to the next service, which is the clinical RNA sequencing. Now, I will present this robust and comprehensive transcriptome analysis of human RNA isolated from FFP or other low quality clinical samples that show degradation. So it gives you like exceptional coverage, focusing in very restricted areas. So it maximizes the discovery power at reduced sequencing depth by targeting these transcriptome coding genes. And in addition, we need a very low total amount of only 200 nanograms of total RNA. In the table, you have more details on these requirements for the samples to be sent to us when you use this service. And so you have also the amount, volume, concentration, and the preferred sample quality and collection and the transportation methods. So giving you like a wider overview on the RNA Stick service, as I mentioned, it gives like a coverage in the expressed genes. So with this, how do we attain this? So we use a very specific strategy for library preparation. So instead of having the typical poly-A capture, we use biotin labeled probes that will be then captured using streptavidin. So this is this strategy is based on TrueSeq RNA exome kit, and it provides a very high quality data. So for this service, aside from having this very good capture strategy for these low quality samples, I want to stress that it only takes 17 calendar days from RNA sample receivable to the release of your raw data. Also, we can provide to you like optional bioinformatics analysis and definitely provide high quality data. So you can see here in this table below how our performance and high data quality metrics are summarized. So for different parameters, when you compare for these type of samples coming from FFP or low quality clinical samples, we see that not only clean data is much higher, the error rate is lower, and we have very good score for Q30 quality value, and a very low presence of rRNA compared to the classic poly-RNA-seq. So here in these other graphs, you can see in a more graphic way what I was mentioning. So you can see that the error rate stays low all along the reads for TrueSeq RNA sequencing or clinical RNA sequencing service. And also that the presence of in the raw reads, the low quality reads is just basically almost absent, it's just very low. And in terms of the genomic regions, it's pretty much like all in exonic regions due to the characteristics of the capture during the library preparation process. So aside from obtaining this high quality data, we can go one step further. And now I will present here the bioinformatics workflow. So after obtaining the clean data for your samples and performing the data quality control and getting this clean data, we would do the mapping against the reference genome, and we can perform standard analysis, getting the gene expression quantification and also differential expression analysis. So with this, we can offer you enrichment analysis to find the different gene expression pathways or biological functions that can be significantly associated with your differential expressed genes, such as using gene ontology, KEGG, or reactome. You can also perform protein analysis using string, and also provide you with function annotation. For the advanced analysis, we can also include the splicing analysis to detect the different types of splicing, which is very relevant in oncology. And also, we can give you SMP and indels analysis, and finally, fusion analysis. Fusion analysis also are very relevant in the oncology field, since, for example, the BCR-ABL gene fusion in chronic myelogenous leukemia is one of the most famous gene fusion events, and you can see how this might be relevant also for your research. So moving now to our bioinformatics solutions, Novogene Antigen 2.0 is one of our most preferred services as bioinformatics. And we can, with this robust and comprehensive bioinformatics service, we use the whole exome sequencing data and mRNA sequencing from our paired tumor normal samples and offer you with comprehensive results that allows us to predict all types of HLA-1 and 6 types of HLA-2, identify 3 types of genomic abnormalities, SMB, indels, and fusions, and their expression level. And also we can provide you with results on TMBs, SMB phasing, and antigen affinity, which eventually will help you to identify the tumor-specific neoantigens. Related to this, you can see on the image on the right-hand side in this figure, the tumor-associated proteins are especially expressed in a high level in the tumor and lower in the rest of the individual, as you can see with blue in the surrounding in the individual. So here would be the tumor, and this is the rest of the body where this would be expressed. However, for the TAA, which is the TSA, which is the tumor-specific antigen proteins, these are only expressed in the tumor, and therefore, since these neoantigens are truly tumor-specific antigens, and they are expressed in here, but not in normal cells, they can be ideal targets of immunotherapy, making, for example, PD-1 blocking antibody be a very helpful instrument. So related to this novel neoantigen pipeline, we can provide this service in 3 working days after your raw data is available. And we would be performing HLA typing, and then also detecting the somatic variations for both DNA and RNA. Also, that will be processed for antigen identification. And then also this pipeline allows the identification of these mutated peptides and the selection of the expressed peptides, and to finally perform the prediction of neoantigen MHC, well, like major histoability complex binding, to determine the potential candidate neoantigens. About the performance of this service, it's really highly reliable, and we obtain the coincidence rate of HLA-1 and HLA-2 molecular typing of 100% and 77.5% respectively, and overall coincidence of 91%. Also, the validation results show really a good performance of this pipeline, showing it has been very rigorously validated, and because it shows these immunogenic peptides that are including 5 HLA-1 binding peptides and 3 HLA-2 binding peptides, showing that have been validated and accurately identified. And so after this, I will move now to the clinical pipeline, which we want to define with like very characteristic words associated with it, which is fast, as I previously mentioned in all the services that we offer in our clinical service. It's consistent, which means that we can provide reproducible results every time. It's verified, that we can have done processing like thousands of samples that provides you high quality performance metrics, and it's traceable all the steps along the way from your project. So related to this, here you have the clinical pipeline, how it goes from sample processing, library construction and sequencing, followed by the bioinformatics analysis, and finally providing to you the project report and the deliverables you have spoken with us. And I want to stress and highlight that all the steps are double checked by laboratory staff to make sure that it goes through a rigorous process. Also that the report is reviewed and signed for a clinical lab member and our bioinformatics engineers. Also that there is an internal quality control for clinical samples before data release. So there is always this last check, making sure that the sequencing results are checked against sex identification. And finally, we proceed a vertical check to confirm traceability. And this is done by the clinical lab manager. So with this, with all this information, I hope you have seen that Novogene can provide you a very good technical support assistance along the project, and that we have skilled laboratory staff, as shown our performance metrics, that we provide high quality data, very fast turnaround time from sample receival until we give you the raw data, which is like normally 15 days or below. Also, we have certified laboratories all across the globe, and we can provide you with customized safe data delivery according to your requirements. Therefore, we believe and we want to be your trusted sequencing partner, and we are ready to accept all your projects and assess you for the best strategy to pursue your goals. So with this, I want to finish my talk and thank you everyone for your attention. And now we'll move to the questions with Marco.
Thank you very much, Aina, for your presentation. It's always a pleasure to hear you. And I'm sure that our audience will have a real clear idea really clear idea of all our clinical services, but some questions have arisen as well. So I would like to start with Vivian. She is asking if we can sequence FFPE samples, and how about tissue samples for whole exome sequencing?
So yeah, definitely we can sequence FFP samples, either being already extracted, or they can send the samples to us and we would do the extraction. In case of tissues, I mentioned only the whole blood and also the saliva, but of course there's a wide range of tissue types that you could be getting from your patients or individual. So we can also provide you the details for this if you were sending us other types of tissue. So definitely we can do this.
Great, thank you very much. As well, also Sijuan is asking regarding NovoPM 2.0, shall we prepare control samples, for example, tumor and control from blood? If yes, shall we sequence both of them at the same coverage?
So, I mean, ideally, when you want to know the somatic variants for your samples, you would send a paired sample so that we compare your tumor sample to the reference from the same individual. However, we have an in-house algorithm that if you cannot provide these paired samples, we can run this with this algorithm that has a very high performance and give you also the somatic variants for this. Also, which was the second question, Marco?
The second question is if we shall sequence both of them at the same coverage.
Okay, so as I mentioned before, depending on the sample type, we have different recommendations. So for example, for genomic DNA, our recommendation is from 12 gigabases, and CT or CF DNA is 16. But for control, we can have a much shallower sequencing with 1 gigabase. It already gives us like 100x coverage, and this can be already being used as a reference. So there's no need to go in the same depth. But of course, these are our recommendations. So you can always go farther and have a higher coverage. But at least to have the great performance, as we've shown with our performance metrics, we recommend this output as a minimum. Yep.
Amazing. Thank you very much. And now Dr. Lee, she's asking, what type of samples do you typically process for exome sequencing? For example, peripheral blood or buccal swabs. Do you also provide extraction?
So we can accept either DNA directly extracted, either like genomic DNA or FFP coming, but also from tissues. The more non-invasive like tissues that we receive is like saliva or buccal swabs. Also, we receive blood, but we can receive several different types of tissues. So it just like depending on our customer preference, we can do this extraction in-house, and if they need like more details on how much they have to send in terms of volume, concentration, or total amount, they can contact us or the local sales representative, and we will guide them on this.
Great, thank you very much. Another question is from Crispin. She's asking, thanks for answering my question. The first time I contacted Novogene, I was told as much as 1 microgram total RNA was needed for RNA-seq. Has this requirement now changed to 200 nanograms?
So because we might be talking about different products, so RNA-seq, clinical RNA-seq is based in another capture strategy, and as I was saying, with biotin tagged probes that allows to focus in the expressed part of the so in the transcriptome, and this 1 microgram probably is related to our total RNA-seq or whole transcriptome service that allows to detect the different variants, so including short RNAs, long RNAs, circular RNAs. So I think like for this reason, this product requires a much higher total amount. But for our strategy for these FFP extracted samples or low quality samples, with only 200 nanograms, we can perform the service to you.
Okay, great. Thank you very much. Yes, the sample amount is highly dependent on the service we offer, and we need to know all the details, of course. So thank you very much. Unfortunately, we have run out of time today, We hope you enjoyed the webinar and found it useful. Of course, we will send you all an email with the link to the webinar and the recording and the slide deck. And this will also include a short survey. We would really appreciate you taking a minute to complete this, as this survey will help us for future webinars to you and focus on your research targets. You can expect to receive this email on Friday. And in the meantime, please stay in touch. You can follow us on Twitter or LinkedIn, and you can get in touch via our website, novogene.com. We look forward to seeing you all on the next webinar. Thank you very much for your attention.
Thank you.
Bye-bye. Thanks, Aina.
Bye.